Impurity identification is the topic for the present video, and in this video we are going to see when the impurity identification is required, why identification of impurity is critical, how it is done, and what are the possible ways to identify the impurity. So impurity means it is not the part of drug substance or it is not the part of formulation. That means it is not excipient. So whatever impurities are there in the API, that is drug substance or the drug product, these are required to be analyzed, these are required to be listed, and these are required to be controlled. The testing and quantification of the impurity in the drug substance and drug product is very essential, as the impurity provides or impurity levels provides the information about the quality of the product, purity of the product, and safety and efficacy of the pharmaceutical products. The products may be API, may be excipient, or may be the formulation. Because impurity levels are directly linked with the toxicity, and that's why the impurity levels affect the safety and efficacy of the API and the formulation. This impurity may come from the contamination or may be coming from the raw materials or intermediates, catalysts, solvents, and maybe the result of degradation. So based on the source, these may be process impurities or these may be the degradant impurities. So this is regarding the basics. Now coming to the impurity thresholds. So impurity thresholds are given in the ICH guideline for the drug substance and the drug products. Impurity thresholds are mainly given as three thresholds: reporting threshold, identification threshold, and qualification threshold. So threshold is nothing but the minimum percentage of the impurity above which the identification, reporting, or qualification is required to be done. So that is the basic meaning of threshold. Now, as given in the table on the slide, the thresholds are given on the basis of maximum daily dose, whether it is drug substance or whether it is drug product. So as per the ICH guidelines, reporting threshold, identification threshold, and the qualification threshold is given. These thresholds are given on the basis of some toxicological implications. Reporting threshold is the threshold above which, or if the impurity level crosses the reporting threshold, then it should be reported. That means it will be the part of impurity list or impurity profile. If it crosses the identification threshold, then it is required to be identified, and if it crosses the qualification threshold, then it requires to be qualified. So identification means identifying the chemical structure and quantifying it. Qualification means generating the safety data based onto the toxicological studies of the impurities. So this is regarding the thresholds for API. Similarly, ICH guidelines give the thresholds for impurity for the drug products, and these are based on to the maximum daily dose. So first is reporting threshold, identification threshold, and then is the qualification threshold. One thing here is to note that identification threshold is for the identification. That means if the impurity level crosses the identification threshold, then you cannot give the impurity as unknown impurity. That unknown impurity, if it is crossing the identification threshold, then it is required to be identified, and then that identified impurity will become known impurity, and that known impurity will have the limit of the qualification threshold. If the impurity is known and if it crosses the limit of qualification threshold, then it is required to be qualified, and based on to the toxicity and safety study data, then that impurity will have the updated or revised specification. So this is regarding the thresholds. Now impurity identification. Identification is needed when the unknown impurity level goes beyond the identification threshold. For example, if your formulation or drug substance contain the unknown impurity, and if the limit for unknown impurity is 0.2% as per the identification threshold, and if you found that the impurity goes beyond the 0.2% level, then the identification is required. Identification means elucidating the chemical structure of unknown impurity, and identification of the impurity is a very tedious, time consuming and critical activity for analysis of the drug substance and the drug product. Once you identify the impurity, you can say that I have done the identification, and now the impurity structure is known. And after the impurity structure is known, that impurity is called as known impurity. It will have the well defined structure and it will have the IUPAC name. Then if that impurity goes beyond the qualification threshold, then toxicity studies can be performed onto that identified impurity. So identification is also a stage or step in the qualification of impurities, because without identifying the impurity, the toxicity studies cannot be taken ahead. Impurity identification is required for understanding the impurity formation pathway and mechanism of the degradation if the unknown impurity is degradant impurity. Also, after identification, the possible ways to prevent the impurity formation can be studied, and the impurity formation can be reduced or prevented. For example, if the drug substance or drug product is sensitive to moisture, and if that impurity is identified, and if it is known that the moisture is responsible for formation of the particular impurity, then based on its chemical structure, mechanism, formation pathway, the preventive measures can be taken, like improving the packaging, storage conditions, and then that impurity formation can be prevented. Knowledge of the chemical structure of the impurity is essential to assess its toxicological implications and to gain the understanding of its formation mechanisms. So, impurity identification is not only for making the impurity known, but also understanding the degradation mechanism, impurity formation mechanism, and for the impurity qualification. So, this is the impurity identification. Now, what are the analytical techniques? So, the unknown impurities may be present at low levels, and these impurities may be complex. So, analytical techniques which are highly sensitive and which are highly specific are to be used for the impurity detection and for the impurity identification. Mainly the separational techniques are used for separating the impurity. The preparative and semi-preparative chromatographic techniques are used for impurity isolation, and then its characterization are done with the sensitive techniques like HPLC, LCMS, NMR, GCMS, NMR spectroscopy, and many more. Some of the time, software elucidation or software based identification of the impurities is also performed. So this is regarding the impurity identification. So if someone asks you the question regarding impurity identification, so you should have the good understanding about this topic. Impurity identification is the first step in impurity qualification. So I hope you might have got good information from this video, and I request you to do like, share and subscribe to this video, and share the Pharma Learning in-depth channel videos to your friends and colleagues. Thank you for watching the video.